Good day and welcome to the second part of our lecture series on human host and microorganisms. I am Professor Teke Apalata, a specialist in clinical microbiology. So let me share my slide with you. And what we are going to discuss in the second part of our lecture series on human host and microbes, we are going to discuss the clinical implications of the bacterial cell growth. So what it means is that when you are infected by the bacteria and the bacteria starts replicating, that's what we call cell growth, what are the clinical implications? So that's what we are going to see here. So these lecture series are parts of our introduction to microbiology. So what is the bacterial growth cycle? Bacteria reproduce by what we call binary fission. Binary fission simply means, binary fission is simply the process by which one parent cell will divide to form two progeny cells. Okay, now, because once cell gives rise, because one cell will give rise to the two progeny cells, the bacteria are said to undergo what we call exponential growth or logarithmic growth. And if you are dealing with a gram-negative infection, gram-negative infection replicates every 20 minutes. They replicate more faster. So if you have one cell, after 20 minutes, you have two cells. After another 20 minutes, you have 4, you have 8, you have 16. So what we say that they enter an exponential growth phase or a logarithmic growth phase. So because here when you have 1, 2, 4, 8, 16, so 1 is 2 to the power 2 to the power 0, and this is power 1, power 2, power 3, power 4. So this is an exponential growth. So this simply means that one bacterium will produce 16 bacteria after 4 generations. And once gram-negative can replicate too fast, every 20 minutes, Mycobacterium tuberculosis, for example, that cause TB, can only replicate every 18 hours. For one cell to give 2 progeny cells, it needs to take 18 hours for Mycobacterium tuberculosis. And it can take you almost 2 weeks, so 13 days to 14 days for Mycobacterium leprae to replicate. So one of the critical information here is that you cannot send, for example, specimen for culture of Mycobacterium tuberculosis and request you send the specimen in the morning, you want the culture to grow in the afternoon. It's not like a gram-negative bacteria like Klebsiella, like E. coli, like Salmonella. The replication rate is different for those pathogens. Okay, but at the end of the day, they will all enter an exponential or a logarithmic growth phase. So this is what we represent here. This is the growth curve of microorganisms. It is represented in 4 phases. The initial phase here that we call the lag phase. The lag phase, so during this phase, the microorganism is trying to attach, to settle, to mobilize all the nutrients and the resources before that it starts replicating. So here you have the number of colony forming units, okay? And this is the time that it takes, like for gram-negative, they replicate every 20 minutes, okay? So after the lag phase, the bacteria will then enter the log phase or the logarithmic phase or the exponential phase, where they will actually then start increasing in the number as a result of replication. then the time, the frequency of replication will depend on the genus and the species of the microorganism, as we said. Then here the microorganism will reach a level where they cannot continuously grow or replicate, because they have to assess the situation. They know that if we continue to replicate, we will lack nutrients, we will lack resources. So some bacteria have what we call the quorum sensing. They can understand the environment, to say, okay, now let's slow down, let's stop replicating because we are running out of resources. That is the stationary phase, what we call the plateau. So they stop replicating. Then they will enter the decline phase, where some of them start dying and being destroyed. So they enter what we call the decline phase. Okay, so those are the different phases, of the bacterial growth curve. So the phase one is the lag phase. We see there is a vigorous metabolic activity that occurs, but cells do not divide yet. This phase can last for a few minutes to many hours, depending on the species of the microorganism. Then they will enter the second phase, that is the logarithmic phase or the exponential phase, where there is then a rapid cell division that occurs, like for gram-negative, we say every 20 minutes, they start replicating. So this phase is very important for clinicians to understand, because antibiotics, most of the antibiotics, especially the beta-lactams, are highly active against bacteria during the logarithmic phase. So they are more efficacious because during that replication, the antibiotics are able to do the job, much more effectively during this phase, because they are able to disrupt the biosynthesis processes during the active cell growth. Okay, so the other implication of this piece of information is that microorganisms, for example, that attach, when let's say you put a catheter in a patient, microorganisms that cut on the surface of a catheter, they form what we call a biofilm. They downregulate their biosynthetic processes. It is very difficult for antibiotics to penetrate and kill those microorganisms, because they are not actively replicating. Then the solution for this situation often is to remove the catheter, because the antibiotic will not be helpful. Then microorganisms will reach the stationary phase or the plateau. So here there is a nutrient depletion, or there are many toxic products that are released. So that will then start affecting other bacterial cells, then they start dying. So then the bacterial growth curve will enter the last phase that we call the death phase, or the declining phase, that is marked by a decline in the number of viable bacteria. So there are different situations here. If you, during a situation where someone is infected by the microorganism, you have the bacteria in your body, they are not running out of resources because you keep eating, you keep supplying them with resources and nutrients. So they have what we call they can continuously replicate because of the continuous supply of the fresh nutrient to them. And you also assist them in removing waste. So that's why the infection without treatment will continue. Microorganisms will continue replicating. The patient will undergo septic shock and die, But in environmental, in the lab, for example, when we are using Petri dishes to culture microorganisms, that environment is restricted because there are only a minimum of nutrients that we put in. There are no additional supply of those nutrients, and there are no removal of the waste. That's where the typical growth curve happened. because there is no continuous supply of the fresh nutrients. But there is another important information here. There are environments that are partially restricted. And partially restricted simply means those environments, the supply of the fresh nutrient is done, but there is a limited removal of the waste. And those type of environments are like abscesses. You know, a patient who has an abscess, you know, the abscess, there is the microorganisms that are found in the abscess. You know, it's very difficult for the antibiotic to go and kill those microorganisms inside the abscess unless you do an incision. Why? Because although there is a supply of fresh nutrients there, but there is no removal of the waste. So the microorganism will not also replicate until the end. So the first example resembles tissue infection, you know, and the second one is a routine lab culture, as I said. And the last one is a situation where there is a pus collection, like an abscess or an empyema situation. Okay, but there are a number of factors which affect the bacterial growth, as we have shown it. You know, among the factors that affect the bacterial growth, we have the nutrient, you know, the type of nutrient, we have the temperature, we have the humidity, we have the pH, we have the oxygen availability, so the partial pressure of oxygen, and we have the CO2 availability. So all those are important factors which affect the bacterial growth, whether it's in our body, whether it's in the lab, whether it's in which environment that we illustrated. So those factors play an important role. Let's look at the nutrients. We have the macronutrient and the micronutrient. You know, all those nutrients play a major role. You know, we have the major nutrients like your carbon, your oxygen, your nitrogen, your hydrogen, your phosphate, you know, all those, those major nutrients are derived from common organic substrates, and they play a major role, you know, in terms of replication of the microorganism. But we also have the micronutrients, and like your ions, your vitamins, you know, they play a major role in terms of as a growth factors. Okay, so let's look at one thing, let's look at the ion, you know, as a micronutrient, the impact of ion acquisition, you know, in terms of bacterial growth and their clinical implication. So we know that iron is an essential nutrient for microbial growth. So microbes also need iron for their own replication. And you as a human host, you also need iron, okay? So the microbes need iron, which by virtue of its low solubility at biological pH is not easily available. So the human host usually has a mechanism, a way of controlling the concentration of ions in our body. So we have, so it's very difficult to have a free ion moving in the body because we have ion binding proteins. We release what we call ion binding protein like transferrin, lactoferrin, ferritin, hemosiderin. Those are ion binding protein. So the reason the body release the ion binding protein because they bind to the free ion. You only have very few amounts of ion that can be moving around, because if we have enough of those, the microorganism will use them, capitalize in those free ion to start replicating and cause infection to us. So the body is also very clever to release ion binding protein to minimize the amount of free ion in circulation. So iron is a very critical nutrient. So then what microorganisms do? Microorganisms will not give up. They come out with different strategies because they want to get ion free from the host, from the human host. So some of them, they will then produce what we call siderophore. you know, so they produce low, they might come out with siderophore that will help them, you know, to take ion from the human host, you know, to take ion from the human host. So those have low molecular weight, ion-specific ligand. They have more affinities with ion than the ion-binding protein from the human host. Okay, other microorganisms will release certain toxin, and those certain toxin will then be released in your body. They will force your body, they will force the ion binding protein to release the ion. And an example of this is Corynebacterium diphtheria that release an enzyme called a toxin called diphtheria toxin. It's also E. coli that can release the alpha hemolysin. So that will force your body, your cells, to release ion. Then those microorganisms will capitalize in those ion. Okay, so while the host tries to limit infection by lowering the ion in circulation, there is adaptation by the pathogen which then try to increase their virulence factors to cause damage to the host so that they can take advantage of that ion to help them to replicate. So beside the nutrient, the pH play also a major role in terms of bacterial replication. Okay, so we know that we have bacteria that are called acidophilic bacteria. You know, an example of that is Lactobacillus acidophilus that's found in the vagina of women, you know. So these lactobacilli play a very important role because its presence maintains the acidic environment in the vagina. It keeps the vaginal pH, it maintains the pH of around 4.2. And that is very important to protect, you know, that's the vaginal cavity, free of other infections, other conditions. So it maintains that low pH. Okay, we also have what we call neutrophilic bacteria. Neutrophilic bacteria are the bacteria that can survive at a neutral pH, that is a pH of 7. And example of these bacteria are like your Escherichia coli and your Staphylococcus aureus. Okay, then we have the basophilic bacteria that enjoy a very high pH, above 7.5. You know, an example of this is Vibrio cholera, is a basophilic bacteria. So you will see that different bacteria, they require different pH condition in order to replicate and survive. So pH is very important. Another important thing in replication of the bacteria is what we call the oxygen. So oxygen use is very important. Now, when it comes to oxygen use, we have microorganisms who can survive in the presence of oxygen. So they take advantage of what we call oxidative pathway. Oxidative pathway use the Krebs cycle or the TCA cycle. You know, so what it only means is that those bacteria at the end of the oxidative pathway, they are able to generate energy, and they generate 38 ATP, you know. So they generate a high amount of energy for their survival, you know. So now to be part of the oxidative pathway, the microorganism should be able to use inorganic electron acceptors. So they are able to use oxygen, or they are able to use nitrate as a final electron acceptors. And they also use cytochromes for them to generate that large amount of ATP. But the others who are not able to use oxygen as the last electron acceptors, they undergo fermentative pathway. You know, so those are only able to generate 2 ATP. So they can still survive, but with very minimum energy because they cannot produce more than that. You know, so they don't use oxygen as the last electron acceptor, but they use flavoproteins. They use flavoprotein. They don't use cytochrome, but they use cytochrome-like molecules. So oxygen is very important for bacterial growth. So that's why we can have aerobic bacteria, or aerobic metabolic bacteria. So they use oxygen as the final electron acceptor. They generate 38 ATP. We can have anaerobic bacteria or anaerobic metabolism. So they use other inorganic molecule as electron acceptor, like nitrate and sulfate. They can still generate more ATP. And we have other type of anaerobic bacteria, especially the strict anaerobic bacteria who rely on fermentation, and they use flavoproteins as a final electron acceptor. They can only produce 2 ATP. So oxygen use is very important in terms of growth of microorganisms. So those that rely on oxidative pathway only are called aerobes. Those that rely on fermentative pathway only are called anaerobes. And you find those that rely on both pathways are called facultative. Facultative microorganisms can switch. They principally behave as aerobes, but when the aerobic conditions are difficult, they can switch to behave as to use fermentative pathway. Example of those microorganisms, the strict aerobic bacteria are your Pseudomonas aeruginosa, is an example of aerobic bacteria. Facultative bacteria are your E. coli, Staphylococcus aureus, Mycobacterium tuberculosis. Mycobacterium tuberculosis is not a strict aerobic bacteria, it's a facultative bacteria, And we have anaerobic bacteria like your Bacteroid fragilis, Clostridium tetani, and other Streptococcus pneumoniae. So those are also anaerobic bacteria. So some are strict and others are non-strict, okay? So non-obligate. But the most important information to remember is also about oxygen toxicity. Oxygen is not always a good thing for microorganisms. For some, it can be very toxic, because oxygen acts as the hydrogen acceptor. We say that it acts as the last electron acceptor in the final step of energy production, when microorganisms are catalyzed by cytochromes. So because the use of oxygen generates 2 toxic molecules, that use of oxygen generates 2 types of molecules. They generate hydrogen peroxide, that is very toxic. They can also generate free radical superoxide, So those molecules are toxic. So to be able to use oxygen as a last electron acceptor, the bacteria should be able to be equipped with enzymes that are able to detoxify those molecules, Otherwise, the microorganisms cannot replicate or survive in the presence of oxygen if it does not have, the necessary enzymes, to destroy the hydrogen peroxide or radical superoxide. So this is very important, So the superoxide dismutase, so the microorganism should have the first enzyme called the superoxide dismutase, because the superoxide dismutase is able to destroy the hydrogen superoxide, because when in the presence of oxygen, when the bacteria use oxygen as the last electron acceptor, there is a production of hydrogen peroxide. So the bacteria need to have a superoxide dismutase to destroy hydrogen peroxide in order to survive in the presence of oxygen. Others can produce what we call catalase, because catalase are very important, Catalase are very important because when you have hydrogen peroxide, that gives water plus free oxygen. So it's because of the enzyme called catalase. So a bacteria needs to survive in the presence of oxygen, be able also to have those two important enzymes, in order to destroy the toxic products that are generated. So this is what I illustrate here about oxygen toxicity. So here you have a bacterial cell, especially anaerobic bacterium. Anaerobic bacterium that is not able to survive in the presence of oxygen. And we will see when anaerobic bacteria can be able to survive in the presence of oxygen. Now, this is an anaerobic bacterial cell. We say that anaerobic uses flavoprotein as a last electron acceptor in the process of generating ATP or energy. So it uses usually, flavoproteins as a last electron acceptor so that there is two molecules of ATP that are generated. But then what happens is that in the presence of oxygen, when you have an anaerobic bacteria, in the presence of oxygen, what happens is that the electron, the affinity of flavoproteins to the electron is reduced. So the electron will be more attracted to the oxygen than they are naturally attracted to flavoprotein. Okay, so those electrons will then be attached with the oxygen, not with the flavoprotein. Then when they are attached with the oxygen, they produce the superoxide that are highly cytotoxic. Now, anaerobic bacteria don't have superoxide dismutase enzyme. And as a result, they cannot get rid of the superoxide. So superoxide will then turn against anaerobic bacteria because anaerobic bacteria does not have a superoxide dismutase enzyme. So as a result, the bacteria will be destroyed. It will die. Okay, so that's why anaerobic die in the presence of oxygen because of the production of a cytotoxic superoxide. And those bacteria, as contrary to aerobic bacteria, the anaerobic bacteria don't have superoxide dismutase enzyme to protect them. Okay, so how do microorganisms cope with oxygen? Aerobes have no problem. Facultative microorganisms can just switch from aerobic metabolism to fermentative metabolism. Okay, but strict or obligate anaerobes are the ones that have problems because they cannot be able, to produce enzyme that can protect them against the cytotoxic products that are generated during this aerobic pathway. That's where the problem is. But then the question is, and we have also another group of pathogens that we call the aerotolerant. The aerotolerant are the microorganisms that are able then to produce superoxide dismutase and to detoxify the environment and survive in the presence of oxygen. But the question still remains. The question that I should ask is, how then some anaerobic bacteria survive in the presence of oxygen? Because like on our skin, there are anaerobic bacteria. There are anaerobes. In our mouth, between the teeth, there are anaerobic bacteria, and there is oxygen there. Now, how do you explain that we have anaerobic bacteria that can survive in the presence of oxygen? The answer is simple. To understand the answer, you need to understand what we call the redox potential. So redox potential is an environment that consists of a mix of both positively charged and negatively charged ions. Now, depending on the amount of ion that is prevalent, that environment can be either positive or negative. So the redox potential can be positive or negative. And the redox potential is expressed in millivolts. So it can be positive, negative, or neutral. Okay. So this is how it is. So in our body, different parts of our body have different redox potential, okay? So there are places where the redox potential is very high, around plus 10 millivolts, very high. And there are areas where the redox potential becomes neutral and it starts decreasing in minus 11 and minus 40, So those areas with very low redox potential are areas where anaerobic bacteria can survive. So anaerobic bacteria can survive in very low redox potential area. Why? Because in those low redox potential area, there is no transfer of electron to oxygen. So the electron will only stick with their affinity with the flavoprotein. So the electron will not be attracted to oxygen. So there will not be formation of cytotoxic products. That's why some anaerobes, like on our skin, we say in our mouth, you have some anaerobic are able to survive in the presence of oxygen because those areas are very low redox potential areas, areas where electrons are not attracted to oxygen. So oxygen cannot play the role of the last electron acceptor for the energy generation. So the electron will only be attracted to flavoproteins. That's why you have anaerobic bacteria that are able to survive in the presence of oxygen. So this very low redox potential area is a reflection of some of the parts of our body. Okay. So let's look at the host-pathogen interaction then. We can have what we call the saprophyte. Just very few definitions. The saprophytes are microorganisms that are usually non-pathogenic. It means that they are not able to harm the human host. And usually their natural habitat is dead organic matters, okay? Then we have the parasites. The parasites are usually unicellular or metazoal organisms living in or on an organism of another species. So the parasites are actually microorganisms that can live on the expense of the host. So they take everything from the host and they don't give anything in return. Those are parasites, okay? So they survive on the expense of other microorganisms. Then we have what we call the commensals. Commensals are normal flora in our body, normal inhabitants, whether you find them on the skin or mucosa, you find them in the body, So they actually live in a very good relationship with the host. They take shelter, food, nutrients from us, but they also protect us in return. So we will see in the next part where we will discuss about the normal flora, you will see that those pathogens are able to give back in return. Then we have pathogenic microorganisms. And there are different types of pathogenic microorganisms that we will try to study. But to understand the pathogenic microorganism, there are two important key information to know. One is what we call the pathogenicity. So pathogenicity is the capacity of a pathogen species to cause a disease. Then there is something in it that we call virulence. When we speak about the virulence of a microorganism, virulence is simply the sum of the disease causing properties of a strain. So what is it that in that bacteria that is actually capable of causing the disease? So all these properties of the bacteria that are able to cause the disease is what we call virulence. But the pathogenicity is actually the capacity of a pathogen to cause the disease. And what is there that is able to cause the disease is reflected as the virulence. So the pathogens are grouped in what we call primary pathogen. First, we have primary pathogen. And primary pathogen are divided in what we call professional and semi-professional. It's just like in a soccer team where you have players who are playing in a bigger club. They are professionals, So those professional pathogens, primary pathogens, are able to cause disease in healthy individuals. So those are the pathogens able to cause the disease in healthy individuals. And usually they are almost exogenous. So they are almost from the outside. They cannot be commensals because they cannot survive with you. They cannot be commensals. Professional pathogen cannot live in good relationship with you. They are there to destroy, even in healthy people. Then we have the semi-professional. The semi-professional are those that cause disease in individuals with sometimes slight immune impairment. Often they cause localized infection because of maybe you have a broken skin, it's a slight reduction in the immune system. They can then take advantage of that to cause an infection. Those are called semi-professional. And they are often endogenous, so they are coming from the host to cause the problem. Then, contrary to the primary pathogens, we have secondary pathogens. Secondary pathogens are simply what we call opportunistic pathogens. So those are able to cause disease in immunocompromised individuals. They can be endogen or they can be exogen, But the problem is, on their own, they cannot be able to cause an infection. They can only cause an infection in immunocompromised patients. Okay, so we say that a pathogen is an organism that has the potential to cause disease in another living organism. And we say that the virulence is a semi-quantitative measurement of the aggressiveness of a pathogen. So what is there in that pathogen, in that microorganism that can cause the disease? So it's an aggressiveness of a pathogen. There are pathogens that are highly aggressive than others because they are well equipped with a different virulence factors. So virulence differs between and within species, even within the same species. Let's say you have genus Staphylococcus, then you have different species. Staphylococcus aureus, Staphylococcus epidermidis, Staphylococcus saprophyticus. Even within the same species, those microorganisms can also have different virulent factors, okay? So that aggressiveness of pathogen, their virulent factors, make the whole difference whether you are going to develop a disease or not. Remember, your body is living in a balanced state where you are healthy. So you are covered by microorganisms that we say commensals. So the microorganisms are there, but your immune system is also there protecting you against some of the pathogen, the microorganism that you can come across with, except maybe if it's a professional pathogen, but you are living in that balanced state. But here, depending on the virulence of the microorganism, because that's what we call aggressiveness of the pathogen, especially the professional pathogens. If you are attacked by those pathogens, then what will happen? You develop a disease because then their weight increase among your immune system. Your immunity is not able to fight those microorganisms because of their level of aggressiveness. You develop a disease. Okay, so that's what happened. Now, there are a number of virulence factors that we should remember, and they play important role at each stage of an infection. The first stage of an infectious disease is its adhesion, where the microorganism is trying to adhere, to attach to the cells. Adhesion. Adhesion is the initial event in the pathogenesis. So it consists of attachment, it's attachment and fusion, So this attachment is not by chance. It depends on specific adhesins, like we spoke about Fimbriae, pili, or other colonizing factors that are able to adhere, to facilitate the adherence, the adhesions, to help microorganisms attach. Then the second phase after attachment and fusion, there is now what we call invasiveness. Invasiveness is the second phase. So this refers to the ability of a pathogen to spread in the host tissue after, in order to establish an infection. So many microorganisms will use different enzymes, different toxins to invade the tissue, okay, to be able to cause an infection. Then there is also the toxigenicity, where you can have, for example, exotoxins that are released by some bacteria. Exotoxins are heat labile proteins, so those are proteins that can be destroyed by the heat, by high temperature, okay? So that's why when you have your food, you put it in a microwave or you warm it, even if they were exotoxin, the toxin that are released, especially if your food is contaminated maybe by Staphylococcus aureus or Clostridium or whatever, but they are heat labile. So when you warm them, those proteins can be destroyed. So exotoxins are heat labile protein which are secreted generally by gram-positive, okay? But endotoxins are heat stable. So they resist the heat. In fact, when you are heating, then those bacteria are being killed, but they are releasing toxin that resist. And you can still warm your food and still develop an infection, okay? So they are heat stable lipopolysaccharides, which form an integral part of the cell wall. Especially, they are released by gram-negative bacteria. Okay, so the endotoxin are integral parts of the gram-negative outer membrane. Other virulent factors can be the plasmids, because they allow the transfer of mutated genes or transfer of resistant factors. You have the capsule that protects the bacteria. Maybe a bacteria is taken up, swallowed by a macrophages, then it releases a capsule, and the macrophage is not able to destroy that bacteria, and the other release various enzymes. We will see enzymes released by Streptococcus pyogenes, by Staphylococcus aureus. We will study those microorganisms in detail and to understand different enzymes that are released. Okay, so to end the second part of our lecture series, on a human host and the microbes interactions and the clinical implications, we have a self-assessment question 3, because in the first part of the lecture, we have 2 self-assessment questions. So this is the question. Some bacteria are obligate anaerobes. So that's what we studied. Some bacteria are obligate anaerobes. Now, which of the following statement best explains this phenomenon? What makes them obligate anaerobes? Okay, 1. So first, get 30 seconds to think about this. Obligate anaerobes. We spoke about the physiology of these bacteria. We spoke about strict aerobes, facultative, anaerobes, aerotolerant, So which of the following statement best explain this? A, they can produce energy both by fermentation and by respiration using Krebs cycle. No, obligate anaerobes don't use Krebs cycle, okay? Because they cannot kill themselves, they cannot accept to use oxygen as the last electron acceptor, okay? So that does not describe them. B, they cannot produce their own ATP. No, this is also wrong, because we say they can produce 2 molecules of ATP, but those that use Krebs cycle can produce 38 ATP, but those obligate anaerobes can produce 2 ATP. So B is wrong. C, they do not form spores. No. Okay, so they are even well known by their capacity of forming spores. So that is wrong. D, they lack superoxide, dismutase, and catalyze. Okay, and the E, they do not have capsule. No, some have capsule. So the answer is D, they lack superoxide, dismutase, and catalyze. Remember, the reason they are killed by the oxygen because they lack superoxide dismutase and catalase that protect them against cytotoxic products that are released during aerobic pathways. Okay, so the best answer here is D. All right, so we come to the end of the second part of our lecture series. Please follow our third video, where we are now going to discuss the implications, the clinical implication of the genetic of the bacteria. Okay, thank you. Goodbye.